My name is Warren Wakarchuk. I'm the Associate Scientific Director of GlycoNet and a faculty member here at the University of Alberta. And it's my pleasure today to introduce our speaker, Dr. Lori Yang. Dr. Yang got her bachelor's degree in chemistry from the University of Virginia and then went on to get a PhD in molecular biophysics and biochemistry from Yale. Her expertise is in protein engineering, molecular evolution, and flow cytometry. Currently, Dr. Yang is the Chief Executive Officer and Chief Scientific Officer of the company Lectenz Bio, which I have to say is a very interesting company. As somebody who used to work on glycosidases, to see that they can be turned into lectins is a very cool thing. At the moment, Dr. Yang heads Lectenz's platform technologies and corporate partnerships. And the company itself was part of Johnson & Johnson Innovation Labs in San Diego. The company is now an alumnus and based at the San Diego Science Center. And I think for those of you who are interested in sort of 21st century tools for studying glycoconjugates, this is going to be a very informative seminar. And with that, I will pass it on to Lori. So welcome, Lori.
Thank you, Warren. Thanks very much for the kind introduction. And thanks to Ali and the GlycoNet organizers for giving me the opportunity to present this webinar. So just a little bit about Lectenz Bio. Lectenz Bio is an academic spinoff of the Complex Carbohydrate Research Center, or CCRC, at the University of Georgia, specifically of co-founder and president, Professor Rob Woods' academic lab. So this is very timely as he gave last week's GlycoNet webinar on modeling glycosylation of the SARS-CoV-2 spike protein. We are headquartered conveniently next door to the CCRC at the UGA Innovation Gateway Incubator. And as mentioned, we have a satellite facility in San Diego where I and a couple other scientists are based. Though not right now, I've been based at my dining table for the last 6 weeks. But actually, we are still open for business, though health and safety remains our top priority. We are working remotely as much as possible while maintaining minimal laboratory operations. And if in the lab, we are following all the precautions, working shifts, sanitizing, physical distancing, etc. So what does Lectenz Bio do? Well, we're mostly scientists and researchers like many of you, and we saw a need for products to make glycoscience simple. First, novel glycan detection reagents called lectins that bind glycans or carbohydrates or sugars. These terms are used interchangeably. Second, a novel glycan analysis tool called glycoscience. And finally, we sell a few enzymes as well, but I'll be focusing mainly on our first two products. Let's start with lectins. So what are lectins? Very simply, they are lectin-like enzyme-derived proteins. And as many of you know, lectins are naturally occurring carbohydrate binding proteins, hence the name Lectenz, which is trademarked and our company's namesake. Lectins are engineered using our patented platform technology for computationally guided directed evolution. First, we start with a carbohydrate processing enzyme for which there is a crystal structure and experimentally confirm the mutations that make the enzyme completely inactive. We do not want any residual enzymatic activity, only binding. But a dead enzyme itself is usually a weak binder. We need to improve the affinity with molecular evolution. So modeling of the protein-ligand complex is used to identify sites which are amenable for mutagenesis. That is, the amino acids in the active site that are essential for binding are retained. The amino acids which do not contribute much to binding are chosen for mutagenesis in order to select for other amino acids in these positions that may enhance binding. We create a focus combinatorial library with all those variations. Multiple rounds of selection and amplification are performed using yeast surface display against the glycan target. Then the enriched clones, cyan, are recombinantly expressed in bacteria and the glycan binding is characterized. This process can be iterative, but if the desired properties are achieved, great, we have a Lectenz. So what makes Lectenz so special? A few things. First, because Lectenz are derived from monomeric enzymes, they are monomeric binding proteins, which can have advantages over multimeric lectins in applications such as flow cytometry, which I'll demonstrate later. Second, unlike most lectins, which are isolated from natural sources, lectins are easily recombinantly expressed in bacteria, so they are very consistent. Third, the specificity is defined by the enzyme it's derived from. So our Lectenz engineering platform can be applied to a number of carbohydrate processing enzymes and their glycan substrates. For example, our first reagents are called Cia-find lectins because they demonstrate well-defined binding towards Cia, or sialic acid, Nu5AC, N-acetylneuramic acid, and in symbolic form, purple diamond. The alpha-2,3 specific lectins is derived from a neuraminidase that preferentially cleaves sialic acid alpha-2,3 linked to galactose. We serendipitously discovered a pan-specific lectins that binds sialic acid irrespective of linkage. Here is a model of the lectins binding to sialylated EPO. So how did we characterize the lectins binding? By affinity and specificity. First, we measured the binding affinity of lectins toward glycan controls. We used a real-time analysis method called biolayer interferometry, or BLI. Shown on the right is a representative centrogram for the pan-specific lectins. We can observe the response changes when a streptavidin biosensor with immobilized biotinylated glycan is dipped into a well containing soluble lectins. The lectins begin to bind, as you can see in the association curves. Upon dipping in a well with buffer only, the lectins dissociates. The table on the left summarizes the binding affinities of the Cia-find lectins. The pan-specific has sub-micromolar KDs for all linkages of sialic acid. That's not bad comparable to some lectins. So it binds alpha-2,3, represented by alpha-2,3 siallolactose, three prime siallolactose, alpha-2,6 represented by 6 prime siallolactose, and alpha-2,8 represented by GD3. It also shows no binding to non-sialylated lactose. The alpha-2,3 specific lectins has a preference for alpha-2,3 linked sialic acid over alpha-2,6 and no binding to alpha-2,8 or lactose. So how do we analyze the specificity? Using Glycan microarray screening. The Glycan microarray is a microscope slide which is printed with sialylated and non-sialylated glycans. The Glycan array is incubated with biotinylated lectins followed by a fluorescent streptavidin conjugate. Now this is really a microarray. More glycans are on the actual array, I promise. So here are the results for the pan-specific lectins. Don't try and read the glycans summarized up here. The pan-specific lectins can bind sialic acid of all linkages, including N-glycosyl acid or Nu5-GC, and not to galactose terminating glycans. The commonly used SNA lectin is supposed to prefer alpha-2,6 linkages, and it does, but it only binds a few of the alpha-2,6 glycans. When you compare the two, actually, the pan-specific binds many more of the alpha-2,6 glycans. So moving on to the alpha-2,3 specific lectins, it shows a definite preference for alpha-2,3 linked sialic acid, but not if it's Nu5-GC and not branched or fucosylated epitopes such as sialoleucin A and sialoleucin X. In contrast, MAL2 lectin is supposed to detect alpha-2,3 linkages. Yes, not definitively, but it's still commonly used for this purpose. It binds only a few of the alpha-2,3 linked glycans. If you compare the two, the alpha-2,3 specific lectins is clearly superior for binding alpha-2,3 linked Nu5AC. So, in summary, Cytosine lectins have been evolved to have sub-micromolar affinities and well-defined specificities. Pan-specific lectins binds all linkages of sialic acid. Alpha-2,3 specific lectins is highly specific for alpha-2,3 linked Nu5AC, but not branched or fucosylated epitopes. SNA and MAL2 only bind to some of their preferred glycans. We are extremely grateful to Shani and Vered at Tel Aviv University for performing these experiments. So what can you do with cytosine lectins? Basically, anything you do with any other protein reagent, like a lectin or antibody. We sell them as kits containing their optimal buffer. I will give examples of these 4 applications: Western blot, immunohistochemistry, immunofluorescence, and flow cytometry, but there are many more applications such as chromatography, immunoprecipitation, ELISA, and so on. The first application is Western blot. We know that the well-studied standard glycoprotein fetuin is highly sialylated. BSA is non-glycosylated, and 3' sialolactose BSA and 6' sialolactose BSA are neoglycoproteins, which have those respective glycans chemically conjugated to BSA. And the glycan stain shows that only BSA is unstained. On the, on the left is a Western blot with anti-6 his, with native cytophyne lectins as the probe. The native lectins that we sell are actually his-tag proteins. As expected, the pan-specific lectins binds all the proteins except for BSA. And the alpha-2,3 specific lectin binds only fetuin and 3' sialolactose BSA, and not at all to BSA or 6' sialolactose BSA. We also sell biotinylated versions of the lectins. On the right is a blot showing a titration of fetuin with biotinylated cytophyne lectins as the probes, followed by a streptavidin secondary. The second application is immunohistochemistry. Human colon tissue was stained with biotinylated MAL2 lectin or alpha-2,3 specific lectins. Upon neuraminidase treatment, MAL2 sections showed no difference in staining, while alpha-2,3 specific lectins is significantly diminished, as shown by the decreased brown color. The third application is also immunostaining, but with fluorescence. Human skin cells were stained with biotinylated pan-specific lectins and a fluorescent streptavidin conjugate in this left panel. A sialotransferase inhibitor abolishes sialic acid signal in this center panel, similar to the levels of the fluorescent streptavidin only control in this right panel. And thank you to Michael and Hans at the University of Copenhagen Center for Glycomics for sharing their data. So the final application is flow cytometry with directly fluorescently labeled cytophyne lectins. This is very simple to do with commercial dye labeling kits. MDA MB231 human breast cancer cells were stained with the fluorescent lectins, as seen in the black histograms. When a parallel set of cells was treated with neuraminidase and then stained, the lectins fluorescence is greatly diminished, as seen by the red histograms. We could not get equivalent data for the SNA and MAL lectins because, as you might guess, the cells aggregated. Thanks to Vlad at the CRO, literally next door to us in San Diego, Curline Molecular Services, for providing the cells. Now to switch gears from our glycan analysis reagents to our glycan analysis tool, GlycoSense. So what is GlycoSense? It is a multiplex bead-based flow cytometry assay for glycoprotein analysis. What does that mean? You can think of it as a lectin microarray or lectin microplate, but on microspheres or beads, not to be confused with the glycan microarray that I mentioned earlier. This is the opposite. The lectins, or any glycan binding proteins, are immobilized on a solid support. In GlycoSense, instead of each lectin being spotted on an array or coating a well, each lectin is immobilized to different color beads. Also in GlycoSense, instead of a microarray scanner or plate reader, a benchtop flow cytometer is used. I just described measuring the fluorescence of cells. Measuring the fluorescence of beads is just the same. And the assays have their own differing characteristics. In theory, the multiplexing level of beads could be hundreds, but practically, for our purposes, there's only several glycan features that we're interested in. And the microarrays and microplates have their own level of multiplexing. Each bead is a replicate rather than the number of spots or number of wells. The sample volumes are comparable, but maybe dependent on your instrument or protocol, and the washes may vary as well. The GlycoSense assay takes basically only the incubation time, as the sample run time is minutes, so can be very high throughput with a microplate loader compared to other methods. But the more tools we have, the better to give options for users, be they glycobiology experts or not, who are more hands-on or hands-off in their work, or who are more familiar or have access to different instruments. So what is the typical workflow? This is a schematic of the GlycoSense analysis. Nothing is remotely to scale, sorry, but it is essentially a mix and run assay. A tube of GlycoSense beads, each conjugated to a different glycan-specific reagent, are incubated with a green fluorescent glycoprotein sample. Either it's directly labeled or a labeled secondary is subsequently incubated. And then the sample is subsequently run on this teeny tiny flow cytometer. The beads are hydrodynamically focused by sheath fluid such that they flow in a single file through this flow cell, enlarged here. Each bead is interrogated by a laser as it passes through the cell. The GlycoSense beads have different amounts of red fluorescent dye, so each bead is shown as having a different level of red color. And in this dot plot, each dot represents a bead. The 5 bead sets are observed as 5 discrete populations along the x-axis. Any glycoprotein bound is measured by green fluorescence in the y-axis. Only bead populations 2 and 3 have glycoprotein bound as seen by their high levels of green fluorescence. Also when converted to a glycoprint. So let's walk through this data acquisition. Here's the flow cell again. As the beads flow through, light scatter is measured. This is a bivariate dot plot of light scatter. Forward scatter and side scatter generally measure the size and granularity of cells. And because beads are so uniform, they have a very tight population as seen in the center of the dot plot. Again, each dot represents an event or bead. There would be thousands of dots. A gate can be drawn around the beads. This tells a flow cytometer we're only interested in analyzing these events, not debris from the sample or doublets to be stuck together passing through the flow cell at the same time. So this is light scatter. What about fluorescence? Well, the red fluorescence is measured at the same time. This is a histogram of red fluorescence. Count is the number of events or beads, and because these are multiplex beads, each of the 5 bead sets has a specific amount of red fluorophore. Equivalent numbers of beads in each set are run, say 1,000 each, for good statistics. That's why in this idealized example, the peaks are the same height and width and very well separated in the histogram. It does look much like this in the actual histograms. If you recall the histograms of cell, they were less smooth. Now, what about the green fluorescence? The green fluorescence is also measured at the same time. We can further gate on only the Sia binding bead population using this marker and analyze it independently from the rest of the bead sets. So now we're looking at the green fluorescence of the Sia detecting bead. The green fluorescent glycoprotein does not have terminal sialic acid, so does not bind to the bead. The glycoprotein is floating around the bead in the sample stream, so does not get measured, as the laser excites only the bead. This histogram, now with green fluorescence in the x-axis, shows low green fluorescence for the Sia bead, therefore no glycoprotein bound. Similarly, we can gate on the galactose binding bead population. And here is the green fluorescence histogram of the gal binding bead. In this case, the glycoprotein has terminal galactose, so is bound by the bead. Because the glycoprotein is now in such close proximity to the bead, the green fluorophore gets excited by the laser and the green fluorescence can be measured. This time, we see high levels of green fluorophore on the bead. In other words, a high amount of glycoprotein bound. And so on for the whole bead set. Again, we observe only the gal and GlycoNet binding beads have glycoprotein bound because glycoprotein has only terminal gal and GlycoNet. To observe everything at once, we can combine the two fluorescence parameters into one bivariate dot plot. Again, each dot is a bead, and as before, the green fluorescence of the gal and GlycoNet binding beads are high compared to the others because they have a glycoprotein bound. This can be converted into a simple bar graph or glycoprint. If the plots seem complicated, they are not. You only have to set them up once. And you can also just collect the data and analyze it with free offline software. We can provide templates and protocols for that to help you out. So, okay, but that was a theoretical, uniformly glycosylated protein. The glycoprint will actually represent a mixture of glycoforms. And as N glycans share a common core, we are most interested in the capping. So what can you do with GlycoSense? Well, you can check your protein glycosylation. Second, you can monitor your glycoenzyme reaction, or some refer to it as glycoengineering. And third, you can monitor glycoprotein production. Monitoring works very well because GlycoSense is best at detecting changes. So we've analyzed a number of other glycoproteins, but for convenience in developing the GlycoSense method, we use directly labeled fetuin. As expected, we see high sialic acid signals and low galactose signals. Now, secondary detection also works. If we incubate GlycoSense beads with unlabeled fetuin, followed by labeled anti-fetuin Fab and a Fab fragment because antibodies themselves are glycosylated, we see that they have similar glycoprints. The fluorescence of the secondary detection is a bit higher because the Fab has a higher fluorophore to protein ratio, or F to P. That is, it had higher amounts of dye conjugated to it. So how do we know what we're seeing is real? Well, we monitored glycoengineering of fetuin over time. Labeled fetuin was treated with sialidase for 1 hour, followed by galactosidase for 2 hours. The data was BSA subtracted and normalized to the highest signal for each bead. As expected, alpha-2,3 and alpha-2,6 linked sialic acid signals, blue and green lines, decrease concomitantly with the increase in galactose signal, this yellow line. After addition of galactosidase at the 60-minute time point, galactose signal decreases, and the GlycoNet signal, gray line, increases simultaneously from newly exposed GlycoNet. We have confirmed the glycoforms at these 3 time points by MALDI mass spec. The glycoforms are consistent with what we observed for our glycosense monitoring. Untreated fetulin has highly sialylated glycoforms. With sialidase treatment, those glycoforms disappear and the galactose-terminating glycoforms appear. And after galactosidase treatment, most of those disappear and you're left with terminal GlycoNet. There is one remaining galactose on a triantennary structure. But this validates what we see in the glycosense enzyme reaction monitoring. In fact, galactose is still going down and GlycoNet is still going up at the time the reaction was stopped. Thank you so much to Peristu Azadi and the CCRC Analytical Services Corps at the UGA for the data. So we wanted to apply GlycoSense to analysis of antibodies, but it has been challenging. Why? Because they don't actually look like this. They look more like this. Can you even see the glycans? They are shown in stick representation here. Furthermore, the antibody subclasses respond differently to the assay. We have to test them on a case-by-case basis. Nonetheless, we wanted to try and detect differences in glyco-engineered antibodies. The first example are uniformly engineered IgG1s with a signal from a BSA control bead subtracted. G0F, the blue glycoprint, is essentially all background. GlycoNet is sometimes inaccessible, so hard to detect. G2F, the orange glycoprint, is terminally galactosylated, so you see high signal from the gal detecting bead. SA3, the gray glycoprint, has only alpha-2,3 linked sialic acid. Thus, the Sia alpha-2,3 bead has high signal, but the Sia alpha-2,6 bead does not have signal. I'll come back to this in a minute. M5, the yellow glycoprint, has mannose, so it has signal from the mannose binding bead. In the next example are uniformly glycoengineered chimeric antibodies, that is rituximab. Thank you so much to Lai Shi Wang at the University of Maryland for providing these samples. They are chimeric antibodies, so mouse variable region, human constant region, and instead of a mannose detecting bead, there is a fucose detecting bead this time. Both glycoforms are fucosylated, as you can see from the high fluorescence of the fucose detecting bead. A reminder, this is not absolutely quantitative because different glycan binding proteins have different affinities for their respective glycans. The fucose detecting protein we know generally gives high signal. Coincidentally, here is G2F again, also in orange as before, and you can also see the gal bead gives high signal. S2G2F, the gray glycoprint, is like SA3 from before, but it has only alpha-2,6 linked sialic acid rather than alpha-2,3 linked sialic acid. Thus, the alpha-2,6 bead gives signal, but the alpha-2,3 bead does not. You remember for the IgG1s, it was the opposite. The alpha-2,3 bead gave signal, and the alpha-2,6 bead did not. So we can detect differences in linkages. So the final application of GlycoSense is monitoring glycosylation of a glycoprotein during production. In this case, the glycoprotein roundabout homolog 1, or ROBO1, which has a single end glycosylation site and is conveniently expressed as a GFP fusion, so the fluorescence readout is built in. And these are glycoprints of ROBO1 GFP taken every day over 7 days of production. The GlycoSense beads were simply incubated with the cell culture supernatant and then run on the flow cytometer. There is no pre-purification of the glycoprotein or enzymatic release of the glycans necessary. So here is the data shown a different way, normalized to protein amount. The most abundant glycoforms of ROBO1 GFP as determined by mass spec include core fucosylated and terminally galactosylated glycans and little Lewis X. So thank you so much to Kellen Moorman and his lab for providing the ROBO1 aliquot, and to him and Paris Duazati for sharing the mass spec characterization data. So, in summary, GlycoSense is an in-process glycan analysis method that is not a substitute for, but is complementary to full characterization methods such as mass spec and chromatography. And it is multiplex. You can run all the beads at once, or mix and match. You can detect glycan features that you want depending on your application. And we can even custom make a bead if there's a particular glycan you're interested in. It's simple. There's no need for release of the glycans from the glycoprotein, or separation, or extensive washing. And it's easy to use, mix and run, does not require highly trained personnel, specialized instrumentation, or software, only a basic benchtop flow cytometer, which some labs have or are easily available at core facilities. And it's fast. It can be monitored in near real time after a short binding step. We are launching this product soon, or depending on the COVID-19 situation, fairly soon. But please do contact us if you want to purchase a trial kit. So our last product line includes enzymes such as the alpha-2,3 sialidase, which performs far better than a competitor alpha-2,3 sialidase. We also sell PNGASF, which performs as well as all the other competitor PNGASFs we've tested. And coming soon are O-glycinase and PNGASF2. Note, PNGASF2 can cleave all N-glycans, including those with alpha-1,3 core fucose. PNGASF cannot. So, finally, I'd like to thank everyone on the Lectenz Bio team. Guess who's based in San Diego? And I'd like to thank all of our collaborators and beta testers, as well as our funding sources, including the NIH GlycoScience Common Fund grant to develop the GlycoSense platform, and as well as the SBIRs to support our product development. Again, we are open for business. Please check out our website or email us. And finally, just contact us if you have any questions or requests. We want to make products that you all would like to use. And again, finally, thank you all for your attention, and I'd be happy to answer any questions now.
Lori, thank you very much for a really great presentation. It's nice to see the development of these tools. And as Lori mentioned, questions can be emailed to her directly. And with that, I guess we'll sign off for today. So thanks everybody for participating in this GlycoNet webinar.